All right, well, thank you for coming to Colloquium today. It's my pleasure to introduce the speaker, Will Jacobs. As you can see, Will is a professor of chemistry at the University of Princeton, a small private university in the neighborhood. And so I've known Will for a long time. Will got his undergraduate degree from University of Virginia, and then he went on to the University of Cambridge to do a PhD in chemistry with John Frankel with a Gates scholarship, and I happened to be at Cambridge around the same time. I arrived a couple of years after Will on the same scholarship in the same group. And then, so afterwards, after graduation, Will did a postdoc at Harvard University, and after that, he landed a job at Princeton. So it's done okay. He's received an NIH fellowship and a career, right? I hope I don't forget any awards. But all right.
All right. Well, thank you so much for the introduction. Had a wonderful visit so far. Enjoyed talking with everyone today. So my talk today is going to be about rational design of complex biomolecular systems. I should say that my group in general is interested in programmable self-assembly, trying to make and understand complex materials by tuning a huge number of different design parameters. And so we do this in two primary applications. One is thinking about soft materials, in particular designing kinetic pathways for colloidal and nanoscale self-assembly. So a lot of our work in this area has been inspired by many people in this department. And so unfortunately, I'm not going to talk about this today. I'm going to focus instead on applications of similar ideas, but in a more biological context. Right, so the basic biophysics problem here is how can you get self-organization inside of living cells through a mechanism of phase separation. And so this is a wonderful example of programmable self-assembly in nature. Right, so the first thing is, what are these biomolecular condensates? The observation of inhomogeneities inside of living cells goes back many decades, but this renewed interest in phase separation in living systems comes from the work of Cliff Brangwyn 15 years ago, when it was first noticed that inside of cells, if you fluorescently tag a particular protein, then you can see that it forms what looks like droplets, and it does so in a way that is quite systematic. And this sort of mechanism, where we're seeing, again, what look like liquid phase droplets, can lead to really beautiful spatial organization. The key thing here is that all of this is taking place in cells that are alive, and these structures are forming spontaneously and doing so without membranes, right? So when you look at this, you should be thinking about different fluid phases that are in contact but are somehow immiscible, and they have different proteins, RNAs, and other biomolecules condensed inside of them. Right, so a lot of the earliest work was trying to show that these have physical properties that we can reconcile with phase separation, as you would see in an inanimate system. So condensates have surface tensions, which means that when condensates of the same phase come in close contact, they can fuse, as you would expect for liquid droplets. It's also possible to observe nucleation and growth, just as you would expect for inanimate phase separation. So here you see that as time progresses in this particular system, a protein concentration is increasing, and it leads to the nucleation of these droplets. But I want to emphasize that these are not simple liquids, right? That there's a lot more interesting physics here than what we know from simple binary mixtures. In particular, these are systems that are composed of a huge variety of different biomolecules. And fundamentally, it's the interactions between these very complex macromolecules that are giving rise to this behavior. So there are an enormous number of different constituents, proteins that may or may not have folded domains, nucleic acids. There's a lot of interest in looking at the interactions between condensates and chromatin, because there are a lot of studies suggesting that these droplet-like structures can be involved in regulating gene expression. And all of this together can lead to a huge variety of different structures. In addition to this, we always have to keep in mind that this is taking place in cells that are alive, and so one should never really expect that these are truly equilibrium structures. And in particular, it's worth keeping in mind that there are always enzymes present, and these can catalyze reactions that are driven by ATP, and thus lead to phase behaviors that are not exactly consistent with equilibrium systems. Right, so there's a lot of complexity here. And so the main thing that I would like you to take away from this is that we can think of biomolecular condensates as a form of programmable soft matter, and programmable in a couple different ways. So one aspect is spatial organization. Right, so it's possible in many of these systems to have co-localization of different types of condensates that form specific morphologies. And this spatial organization is key to their biological function. It's also possible to control these in a temporal sense. So living cells use this to assemble and disassemble these structures, which again don't have membranes, so it's very easy to form them and dissolve them in a way that is important for their biological function. This is also possible to be controlled experimentally. So in this case, the nucleation and then dissolution of these droplets is under experimental control. And a really important aspect is that these various droplet-like structures can be enriched in different types of biomolecules. Right? So the biological function, what is happening inside of this self-organized structure, follows from the types of molecules that are present there and are enriched in this environment relative to what else is in the cell. Right? So this can be important for driving chemical reactions, preferentially in one location within the cell by concentrating enzymes and molecules there. So within this view of programmability, my group has been thinking mostly about design rules for constructing programmable condensates. So we've been doing this in a couple of different ways, thinking about how do you control the compositions of these multiphase structures? How does that connect back to biopolymer sequences? Thinking about the complexity of forming different oligomers, tuning different physical properties and phase boundaries, all by making subtle changes to protein and RNA sequences. Also been thinking about how these condensates can organize into higher order structures, such as these multi-scale structures that I showed before. And also the kinetic aspects or dynamical pathways by which you can form and regulate the assembly of such structures. So today, I'm going to pick out two aspects of this. And so start off by telling you some of the interesting things we found in terms of multiphase organization and controlling compositions in these systems. And then at the end, some interesting things about nucleation pathways in systems that are driven out of equilibrium. So to start, why we care about compositional control. If you, and this review is a couple years old now, so this is not even anywhere complete inventory of different condensates that have been well studied in the literature, but there's a very large number. These things are all mutually immiscible, which means if you bring them into close contact, they don't fuse. And if you pick out a particular one of these structures and ask what is inside, you also observe a lot of complexity there. So here we're looking at partition coefficients, which is simply the ratio of a protein concentration or molecular concentration inside the droplet to in the rest of the cell, either in the rest of the cytoplasm or the rest of the nucleus. What you see is that there are a large number of different protein components in this particular study that are enriched in this one particular structure. And in general, there's not a one-to-one correspondence between a biomolecular species and the structure into which it tends to partition, right? So we can look at some of these particular proteins and find that they might go into a large number of these different droplets under different conditions.
How do they classify the different condensates?
So most of these things are coming from studies that are from a biological viewpoint, so thinking functionally, right? So particularly inside of the nucleus, there's this vast array of structures that are known to have different key functions with regard to transcription, splicing, that sort of thing. And it's only recently becoming clear that some of these things are forming by the space separation. A lot of these have been known to some extent for a much longer time. So the way that we've tried to approach this question of how you can get such complexity from heterogeneous mixtures of biomolecules is to think about the inverse problem. So instead of trying to take this complex mixture and predict what we are going to find in terms of phase behavior, we want to say, let's propose we want to make this phase behavior and then work backwards and figure out what sort of biomolecules could potentially do that. We would want to design a mixture. And to make this highly non-trivial problem a bit easier, what we have done is to utilize an assumption that I think will convince you later on can be extremely reasonable for these systems, and that is to use only pairwise interactions. Right? So that means that that every molecular species can interact with any other molecular species with some effective interaction strength. So this is a coarse graining of all the interactions. Homotypic interactions are between the same type of molecular species, and heterotypic with unlike pairs. And these interactions have a clear physical meaning. Right? So this is an approximation, but these partition coefficients that I'm speaking about, this log ratio of concentration inside of a condensate to what you find outside, when you invoke this pairwise approximation, we're just simply saying that there are some coupling coefficients that describe this ratio in terms of all the other concentrations, all the other species in the system. So the basic idea is that we can use this approximation to try to do this inverse design problem.
Well, those look to me like, the formula trying to interpret it looks to me like those would be chemical potentials. Yep. So is there any functional difference between, is it because it's on a lattice that concentration and chemical potential are the same thing, or?
So it's not on a lattice, right? So no lattice here. This is a matrix of interactions. So this, yeah. So you can think about this. This is my epsilon matrix here, but from one species to another species. Yes, this is making the approximation of the excess chemical potential. The part that depends on these effective interactions is linear in concentrations. Right? And it's a starting point, but it allows us to actually carry out this design problem. Yes.
So on the same equation, can that be used to predict, for example, as LCST or UCST transitions based on the like the left term?
Yes. So the KT part, it can really just be wrapped up into our effective interactions. This is something that you can compute or you can measure experimentally, and if you do that at a single temperature, then that would be the input. Okay, so the basic question is, let's suppose that I have a mixture. I want to design a large number of condensed phases. These phases might have shared components, right? So in this cartoon, each one of these blobs is a molecule, and these are condensed phases indexed by alpha. And this is my design problem. I wish to tune this interaction matrix such that I get these phases. So in terms of thermodynamics, what this means is that we should have a free energy landscape that has many local minima. Each local minimum should correspond to a particular phase. The reason it has to be a local minimum is that we want to make sure that these phases are immiscible. So if these condensates come into contact, they don't fuse. That means they need to have a positive surface tension between them, which corresponds to a free energy barrier between different phases. So this becomes a problem in designing a free energy landscape, right? So as Glenn pointed out, when we actually put this into a simple mean field model, our chemical potential is related to this set of coupling coefficients or pairwise interactions. And we have two types of constraints. So one type is our design, saying that we want to have a certain set of condensates with certain compositions. And then we have these thermodynamic constraints, which say that we need to have local minima, or in particular, this means we have constraints on the eigenvalues of the Hessian matrices at all of our condensed phases. So this is a highly nonlinear problem, but what you can do is a convex relaxation, which means that we're going to make some controlled approximations on these thermodynamic constraints to make the problem a little bit easier to solve. What that means in practice is that the solution space gets a little bit bigger. Okay, so we can imagine that all possible interaction matrices live in some interaction matrix space. And then within that space, there's potentially some solution space. These would be the interaction matrices that solve the proposed design problem. The convex relaxation is a bigger space, and we're going to pick out some solution from within that. Making this problem convex means that we can actually prove whether the solution space exists at all. And so we can say, for a given design problem, is there an interaction matrix that could conceivably solve it? So when we put this idea into practice, we can then ask, in a system with a given number of components, if we have the ability to tune all of these interaction parameters, how many condensates can we possibly design? And so, of course, the answer, yes or no, is there a feasible interaction matrix, depends on exactly which molecules you want to go into which condensates. But there is some typical behavior that can give you a scaling law. So this is the typical number of condensates that you can program. So in general, in a complex system, if you ask me to program fewer than this typical number, it almost always works. I almost always find a solution. If you ask me to program more, usually there's no answer. This scaling law goes as a power law in the number of components that come available to you. And interestingly, this exponent here can be greater than 1, which means that when we're trying to design these local minima on a free energy landscape, it's possible to have a number of condensates that grows superlinearly with the number of components.
So the points on that plot are, so what you do is that for a given matrix, you can determine whether a solution exists.
For a given design problem. So you say which molecules you would like to go into which type of condensates, okay. You prescribe all that, and you say, is there an interaction matrix that would do that? So this is not saying if the matrix is, although we can find that, and I'll show you that, but here this is just saying, is there?
What is the meaning of index 1/2 of this little typical number of condensates?
Yes.
And 1/2. Why is it?
Right. So 1/2 because it means that this is the point at which I have a 50% probability of being able to find a solution. Right. So the actual whether you can find a solution or not is binary. Right. But if you propose many different design problems that all have the same number of condensates, but scrambled which protein goes where, the details of the design problem change, and thus you may switch from being having a feasible solution to an infeasible problem. But this is the point at which you tip over from usually having a solution to none. Okay, so this goes super linearly, right? So at this point, you might be thinking, what about the Gibbs phase rule, right? Which you expect everything only goes linearly. According to this, according to the Gibbs phase rule, right? And the key point here is that we are thinking about making local minima on a free energy landscape. So this means that we don't necessarily have to have all the condensates present interacting with each other at any given time. They just need to be able to be metastable with respect to all of the complex mixture around them. Right, so to show you that this works, we can do a simple lattice model. This is a growth simulation where we have a seed of our target phase, right? So there are approximately 100 different species here, and over 100 different condensate targets. But I'm only using 2 colors. So for this particular seed, a red color indicates that that molecular species should be in the condensate according to my design problem. So as you run the simulation, from this heterogeneous mixture, you're able to grow this condensate with a stable composition because it remains red, right? In a way that depends on the seed that was provided. Right? So again, this is not a violation of a Gibbs phase rule. This is simply showing that local stability is what matters when you're trying to make many different types of phases out of a complex mixture.
If you allow for more than just 2-point interactions, you have a lot more Is that then allow you to have things to grow much faster?
Presumably, yes. The main reason we've limited to this is because all of this is motivated by biomolecules, where you tend to have very short range interactions by comparison to the size of the protein or RNA. But presumably, yes, you could recast all of this in a way that would give you even faster growth of this thing versus this thing.
There must be real examples where, say, you have a nucleotide binding protein that once it coats the genome, it's in a different configuration and then it interacts differently with other repressors or whatever, right?
Yes, indeed. Right. So there are certainly cases where you should have more than this pairwise approximation, but this is a reasonable starting point, and already you can observe some quite unexpectedly complex organization that comes from it.
Does the existence of a solution or not depend on the intensities that it's high end?
Not really. It is very robust to that requirement. It's mostly just in or out because things either need to be attracting or repelling, and that's what it is. And so the theory actually that's given here is that highly simplified version, just saying, I like you or I don't like you. Okay, so at this point, we need to take a step back and ask, oh, sorry.
Sorry, I'm confused because I fail to understand why you would not get an interaction matrix. Like, let's say you want component 1 and 3 to be in a condensate together, you just increase or decrease the interaction energy between them, and then they coalesce, right? That's,
yes.
So what are the scenarios where you don't get an interaction matrix?
So when I say that an interaction matrix may not exist,
yeah,
means that I prescribe, I would like molecule type 1 to go into condensate type A, molecule type 2 to go into condensate type B, and if you ask for too many things, too many constraints, then there may not be a matrix that solves that problem, which means that then all of these things, there are of course interaction matrices, but if you were to use them, the proposed condensates would all mix together.
Okay.
They would not give you the structure that you asked for. Gotcha. Okay, so at this point, you ask, can you actually make these interactions using real biomolecules, right? Because so far we just have these coupling constants, and in particular, in this design space of possible interaction matrices, usually have a highly degenerate space of solutions. Right? So some of these sets of interactions, some of these mixtures, may be easier to realize experimentally than others. Right? So how do you choose the simplest possible matrix? Okay. So here we need to put our chemistry hats on for just a moment and think about what types of actual physical interactions are leading to this thermodynamic driving force for phase separation. Right? And so again, this depends on the particular biomolecular context. But in the case of proteins, typically amino acid properties such as electrophobicity and charge that tend to drive phase separation. For RNA or DNA mixtures, nucleic acid motifs. We can also have protein-protein interactions between folded domains, right? And so in a very general sense, in each of these cases, you can think about a number of linearly independent interaction modes. And so what we should be doing is not thinking about the space of interactions, the space of interaction matrices, but rather the space of these interaction modes. Right? So the key idea is that we should just factorize, and we have our interaction matrix, and if we want to write this in terms of the available interaction modes, then we have a feature-feature interaction, say how hydrophobicity interacts with charge, hydrophobicity with hydrophobicity, and then how those various features go into each of our molecules. Right? So each row of this matrix here would be the appearance of these various features in a particular molecule. And so the complexity of our solution, our design solution, is given by the rank of this matrix here, because this tells us how many different distinct interaction modes we need in order to build the actual biomolecular mixture. And so just to convince you that this is actually a serious consideration, you can take a toy model of hydrophobic and polar beads. Right? So this so-called HP model has been around in the protein folding world for ages. It's basically the simplest copolymer model you can think of. The H's strongly attract other H's. And so if I consider all sequences of a particular length, say length 8 in this case, I can compute the potential of mean force between these two polymers and thus get my net interaction. And actually compute what one of these interaction matrices would be. Right? So all possible 8mers versus all possible 8mers, here are my interactions. And now if I factorize, if I do an eigenvalue decomposition for this matrix, I will find out the number of unique interaction modes. Yes?
And self-interactions are on the upward going diagonal?
Yes, this,
okay.
In this, yes. And so the upshot is that there are only, in this particular common simulation model, there are only 4 non-zero eigenvalues, which means that there's actually very low information content here. Right? So we have a lot of possible sequences, but in terms of these net interactions, there are only a couple linearly independent ways that
so the interactions is you just slide the two.
This is a simulation, off-lattice simulation of essentially Leonard-Jones beads, where we have hydrophobic interactions have been attracted well, and these polymers are fluctuating, right? And we bring them close together, pull them apart, and measure the potential of mean force for doing that. The same way that you would for, say, finding the effective interaction between two colloidal particles or anything like that.
So the polymers can bend whatever way they want. The only constraint is in the sequence.
Yes, I'm changing the sequence, but when I do the simulation, I'm just pulling the center of mass apart to get the effective interaction, right? So this number here is essentially the second virial coefficient, the net interaction that's directly related to that epsilon ij that I showed before. So this is per polymer sequence here, right? I get a single number for how that polymer sequence on average interacts with another polymer sequence if these two chains are in solution together.
2 to the 16th simulations.
There's actually some degeneracy because
right.
there's no head to tail, so
But
that's a lot of simulations.
So do you, so in this case, if you had these, let's say, individual beads which were not connected, would that potential well look the same as if when they were connected? Like, do you account for patterning in some way?
Right. So this is taking into account the patterning. Right. So if you had the disconnected beads have a different potential mean force than when they're together in a polymer chain. But we're not putting anything by hand about how we think patterns matter. This is just what happens in this simple simulation model.
Yeah, but let's say you have a PHP, will the interaction potential of that H with a different H be different if it was like PHH? Like, is that information
So
included?
so there are only two monomer types here, right? So H's interact with H's or H's interact with P's in the same way always, no matter where they are in the sequence. Right? But we string them together into polymers, and then these are the net interactions between the two polymers.
We're saying just things cool that you calculate, but you're not doing a full umbrella sampling calculation. You're doing some kind of, or you are. Okay, so you do. So how much do you need to do to converge this when, let's say, some of the polymers might collapse on their own, but when they're together, they're extended?
It's a lot.
Yeah. But like, do you know, how do you check for hysteresis there?
Yeah. So these short chains are actually very well converged. Right? And we've done this in a couple different ways, different enhanced sampling techniques. We have to say this. Of course, to be very careful about what you consider to be the noise, because you have a random matrix problem here. But yeah.
So the fact that you have these lines means there are certain sequences that interact strongly with many other sequences.
Here.
Yeah.
Yes. Yes.
Is that related somehow to the simplicity? There's just a few of these sequences that interact strongly, and then just a few ionized that dominate.
It's mostly just the way that I have them ordered here. You could rearrange this matrix, and then you wouldn't have lines. I think yes, there's a clear block structure here, but this is just related to the fact that there's very little information in this matrix. It's very low rank. So what we want to do is put this idea into our inverse design approach. We want to look for interaction matrices that are low rank. And we want to find the ones that will be easy to make. So let me give you a specific example of how this works. So suppose we want to make these different condensates. Each one has a particular set of molecules, right? I'm going to call the molecules red, green, gold, and black. I want to make this. Okay? So then we do this optimization procedure, but now we're going to do the convex optimization while specifically looking for low rank solutions. These will be the easiest ones to realize. And so whenever you reduce the dimensionality or you reduce the rank of a matrix, you tend to introduce errors. And so when you simplify your full rank matrix, you have an interaction matrix error here. And what we're able to do is derive a bound that says that as long as your error is below this bound, you're still going to get back to essentially the same composition. But if you are above this bound, then you're going to get errors in your composition. I won't make what I want. And this bound is a function of how stable you make the phases. So there's a trade-off here. If you make more stable, if you want the phases to be more thermodynamically stable, then you can tolerate more errors in your interaction matrices. And so if you actually do this approach, this is a complicated plot, but the key takeaway is that if you only, if you use all four interaction modes, because I have 4 molecules, then you have by definition 0 error. If you use only 3 modes, you have low error, but if I reduce down to 2 interaction modes, then I can't satisfy my bound, and therefore I should not get the right phase-separated structure.
Seems to me that you artificially chose your desirable structure such as to make your life easier, because you don't have any frustration there.
So this is a particular example. Yes, I give you that.
Sure. The problem will arise if, for instance, red wants to be next to yellow in one condensate, but wants to be away from yellow in another.
But that is what's here, right? Red wants to be next to yellow here, and red wants to be near green here, but I don't, but the idea is that yellow should be excluded from condensate number 1. So this,
So that just means that yellow and green hate each other.
Yeah.
That, yes. And so, exactly. And so, this is something that has a solution, but we're trying to now say, how many modes do I need to do it?
Do you agree with me that it's easy to construct an example where the requirements are truly contradictory?
Yes, and so if you construct an example where it's infeasible, then the convex optimization tells you so. We'll say that's why earlier on when we said if you just randomly propose design problems, if you try to put too many constraints, then it becomes infeasible. Right? So this problem has a solution, but we're trying now to find the simplest possible solution. And so
for modes, you mean the rank of the
rank matrix, right? Yeah. So exactly, and so from this point on, I'm going to make the assumption that we're going to treat the number of monomer types, right? So before we had H's and P's in our sequence, so now I'm saying I have 3 monomer types. So now as a second step, I'll just design polymer sequences. I want to factorize this interaction matrix. So I have this low rank interaction matrix, factorize it, and that gives me my polymer sequences. So to show you what this does in practice, the prediction is that these polymers here should phase separate into immiscible phases as shown in the example of the thing that I wanted. So here they are. There are 3 monomer types, gold, yellow, and blue. And then to see if this actually works, we're going to run a molecular dynamics simulation where we do what's called a direct coexistence approach, which means we put our 2 condensed phases in direct contact and also in contact with the dilute phase, and we let the system equilibrate with periodic boundary conditions, and we see if we maintain immiscible structures. So it's hard to see here because of the colors, but in order to match this back to our prescribed design, we're going to introduce an order parameter that's equal to 1 if we have the precise composition that we requested for that phase. So the fact that the bulk of each of these condensed phases has an order parameter equal to 1 tells us that we've made what we want. And here, because we had 3 condensed phases, we have 3 pairs that we need to consider, and this works for all of them. So the upshot here is that this is a simulation test showing that indeed you can get the requested phase-separated structure, 3 different condensed phases with those precise compositions, using only 3 monomer types. In all of our tests, if you use only 2 monomer types, this does not work. Right? So you can look at these order parameters, define an accuracy, and it's a failure if you don't use sufficient number of interaction modes or monomer types in this case. So the key takeaway here is there is a minimal complexity of your sequences that you need in order to be able to build a complex phase-separated system. Right? And in this particular case, the answer is 3. We do this for other cases, right? So just as an example.
Well, here, sorry, here you're making sure that they don't mix. So you're putting them in contact, making sure that they stay, they remain unmixed. Okay.
The key thing here is just checking for immiscibility that these phases don't merge. Here, if you only use 2 monomer types, you find that all these phases merge. Okay. To do this for more complicated examples, for example, you can have more condensed phases than the number of monomer types that are needed, and go through all these tests. And so what this does is it provides a test of our optimization theory, this context optimization approach, telling us the number of monomer types that we need. And this is this post hoc validation of this pairwise approach, this pairwise assumption that we used at the beginning. The reason it works fundamentally is that these are nearly polymer melts, and polymer melts tend to be quite neat. So in summary, we have the ability to design compositionally distinct condensates. In principle, you can make a huge number of them, but in practice, it's usually the amount of chemical information that's in these polymer sequences that really constrains what you can do in practice, right? So with this algorithm, we can compute what you need and then systematically design subsequences. This is something that you can apply to wide variety of different biomolecular systems, disordered protein condensates, nucleic acid condensates, potentially colloidal foldamers even, as an idea. Okay, so in the second part, I'd like to move on to a different aspect of self-organizing condensates. In particular, if you have the ability to create all of these different structures, it becomes important to say, how do I get a particular condensate to appear at a particular time in a particular place within the cell? Because that's what's observed. And so overall, this really boils down to nucleation dynamics. So as shown in this beautiful paper from Cliff Brangwyn's lab 2 years ago, it's possible to track the nucleation of these condensates in living cells. Typically, you have relatively low protein and RNA concentrations, which means that move past the phase boundary into a metastable region where there is a sizable nucleation barrier. So if you watch this movie, you'll see that towards the end, some of these condensates start to nucleate stochastically. And in general, expect that you have a nucleation rate that increases with the concentration of whatever components are inside here. So our basic equilibrium picture of phase separation says that if you control protein concentrations or RNA concentrations, you should be able to tune the nucleation rate of these things, and that indeed that more or less holds true. And so the key idea here is that because we have these highly heterogeneous complex systems where you observe the specific nucleation of certain condensates, this is a key mechanism of spatiotemporal control.
In those experiments, are they forcing the cell to express certain proteins?
Here they're doing a trick where actually they cause certain proteins to oligomerize when the light is turned on. Mm-hmm. And so instead of directly changing the concentration of all proteins, they're changing the concentration of dimers or oligomers, right? But it's also possible to do this with other mechanisms, and something that we're working on is using small molecules to disrupt different interactions in a way that's actually much more physiologically relevant. All right, so just as a reminder, probably not needed for this audience, classical nucleation theory. The basic idea is that as you increase the size of a cluster of this condensed phase, right? So this is like a cartoon of this droplet growing. At some point, you go over a nucleation barrier and it becomes favorable to continue growing this droplet. So in equilibrium systems, we know that this free energy barrier is a non-monotonic function of the size of this cluster or the size of this nucleus, right? And so there's some overall free energy barrier at the top that corresponds to a critical nucleus size. For nuclei that are bigger than that, classical nucleation theory predicts that you should go downhill in free energy and thus grow, right? And the key idea is that in these systems, if you increase the surface tension, then it becomes harder to nucleate. It becomes harder to reach the top of this barrier because the thing that is causing your free energy to go up at the beginning is the cost of creating an interface between the two pieces. Okay. So all of this works reasonably. Yes.
Just a quick question. I mean, how do you know that actually nucleation and growth is the right process compared to, for example, spinodal decomposition?
So mostly because of the very low concentrations, and again, there are almost, in biology, there's probably an example for almost everything, and so there probably is a case where this is driven by spinal decomposition. But in the vast majority of systems where you have relatively low concentrations of these various species, things that are more akin to a nucleation growth pathway seems more appropriate.
So concentration aren't by definition, when you see phase separation, you're supersaturated. So
Yes. Right, so there's some phase boundary, some critical concentration that you have to go past, there's saturation concentration to go past. Once you do that, it becomes possible to phase separate, or whether you actually observe that in practice depends on whether you go over a barrier like this. Only at much, much higher concentrations would you actually hit a spatial regime.
So the homogeneous phase is still there, but it's just metastable. It doesn't become unstable.
Correct.
Yeah, I see. Okay.
So is it, shouldn't that depend on the two rates? One is the rate of growth, and then one would be the rate of spontaneous fusion, which is kind of a proxy for surface tension.
In a sense, yes. So in this old classical theory, right, that the two parameters you have to work with are the thermodynamic driving force, which is supersaturation, and the surface tension that tells you the penalty that you're paying to create a small droplet. You can infer both of those things, I think, from what we were suggesting. Okay, so what we wanted to think about in this case is, in these systems that are living and have enzymes and ATP that can carry out reactions, a lot of which chemically modify the components that are undergoing phase separation, is it possible to observe different changes, different types of nucleation dynamics, right? And with the reason being that that nucleation giving you spatiotemporal control means you have to be able to selectively change nucleation rates for different types of droplets in the system. And it turns out that yes, if you think about systems that are driven away from equilibrium, particularly by chemical reactions, it is possible to introduce new ways of changing nucleation rates. So the basic model that we have in mind is phase separation at a chemically driven non-equilibrium steady state. And the steady state part means that on average, nothing is changing in time. It's non-equilibrium because it will never relax the thermal equilibrium. In most living systems, this is because you have a constant supply of ATP that's always being replenished, and that can provide a chemical potential to drive any reaction that is coupled. So we have macromolecules, solvent, chemical fuel, and enzymes. The simplest scenario that you can imagine is having a phase-separated droplet where we have a system with two types of macromolecules. A phase-separating macromolecule that I'll call B for bonding, and an inert one called I, and they can undergo this unimolecular reaction with some equilibrium free energy difference. So this would be your undriven system where I have chemical reactions with phase separation. So the key idea is in a system that's in non-equilibrium steady state, you can have an additional reaction pathway that's driven by a chemical fuel source. Right, so here our chemical fuel might be ATP, this might be ADP, and it imparts some additional driving force that we designate delta mu. And the key theoretical framework that we're going to use here is stochastic thermodynamics, which says that we're going to assume that the solvent and chemical fuel are always at equilibrium. They provide a bath or a reservoir, but our macromolecules that are undergoing phase separation are potentially driven far from equilibrium. In particular, the populations of these two states could be driven far from equilibrium.
So sorry, when it's inert, it means it just doesn't interact with anything.
Doesn't interact with anything. It will not, far above critical temperature, it will not, critical binding energy will not phase separate.
So that's the idea of some kind of probability, like some modification to the polymer that.
Absolutely. So a key biological example is phosphorylation. There are, it's different in different contexts, but there are numerous examples where if you phosphorylate with kinase, then you can get the molecule to phase separate, and in the absence of phosphorylation, it will not phase separate. So that would be something like this, with this free energy difference getting between two states, the two molecular species.
Does that go? It's only one directional, and that's irreversible. Or
so everything should be reversible. maybe with a very, very slow rate constant, but there is always if this reaction can happen, then there's a free energy difference, and it must be able to be reversed. Okay, so if you put together a lattice simulation to try to study this, the first thing you'll notice is that in a system that's driven out of equilibrium by chemical reactions, you can have steady state fluxes. Right? Steady state fluxes are prohibited at equilibrium, but at a non-equilibrium steady state, they can exist. Right? And so here, we've chosen as a particular example to have the B to I reactions along the driven pathway be concentration dependent, right? So, for example, this could be driven by an enzyme where the enzyme partitions into a condensed phase, right? Because it interacts favorably with the condensed phase, so that B to I interactions are preferentially driven only in the condensed phase, which I'll just refer to as liquid and vapor for simplicity here. Okay, so because of this, B's are converted to I's inside and then tend to flow out. I's are converted to B's, preferentially in the vapor phase, by the undriven pathway and tend to come back in, and you get these sort of fluxes. This is what you should expect for a system that has a non-equilibrium steady state with these sorts of driven reactions. What you might not expect is that there can be enormous changes to the interfaces between these phases. And so here, the simplest thing that you might think to do is to just measure the typical height fluctuations, or root mean squared displacement, of the interface between the condensed and dilute phases, and then compare those to what you should have at equilibrium. Okay? So at equilibrium, if you keep the temperature constant and you just change concentrations, this delta HEQ is a constant. But now, depending on how far you drive the system away from equilibrium, you can depart from this point, this equilibrium point at 0, 0, and have larger fluctuations or smaller fluctuations. And when you see these changes in fluctuations, you should think, okay, there's a change in interfacial tension here, or at least something that looks like an interfacial tension. And so I can change interfacial tensions, then I may be able to change nucleation rates. Okay, so here, going back to this classical nucleation theory picture, you can write down an expression for the nucleation barrier height in terms of things that we can calculate independently in these simulations. Right? So we have to use enhanced sampling simulations to calculate rates, and then compare this to supersaturation that we're able to extract. And at equilibrium, there's a single curve along which everything should lie that's defined by this the temperature. When we introduce these driven chemical reactions that are now far from equilibrium, our delta mu is much larger than KT. Overall, you can fit this to classical nucleation theory, but the apparent fit indicates a changing interfacial tension. So this is, in this case, decreasing, which is consistent with the height fluctuations. So if you put all this together, you can have a new mechanism for controlling a nucleation rate, independently of the supersaturation and the temperature in the system. So the key idea here is that I'm going to drive the system out of equilibrium while maintaining a constant supersaturation and a constant concentration of particles in the homogeneous vapor phase. At equilibrium, if you specify this and this, then for this biomolecular system that I showed you, there's a unique nucleation rate, and that's JEQ. But by driving things out of equilibrium in this way, where we promote the B to I transitions in the liquid phase, you can get a nucleation rate that varies over multiple orders of magnitude with relatively small changes. So this means that you only need a relatively small driving force at the level of these B to I transitions in order to affect a massive change in how rapidly this droplet will nucleate. And this is a completely tunable effect. So if you control this drive, you control where these reactions tend to take place. Are they primarily in the liquid phase, primarily in the vapor phase? You can tune this effective interfacial tension sigma C and T, indicating that this is the effective interfacial tension that's extracted from the nucleation analysis. And all of this can be described quite beautifully by some theoretical predictions that invoke some simplifying assumptions of effective equilibria in systems that are at a non-equilibrium steady state. So in summary, we've seen that it's possible to tune interfacial properties independently of the bulk properties of these phases, independently of supersaturations or molecular compositions, and that this can have massive implications for nucleation rates. In particular, giving cells the ability to change how rapidly things nucleate by controlling where enzymes want to go in these systems. So this is one possibility in these living systems for controlling the assembly of condensed phases, a mechanism that's not available at equilibrium. And I think perhaps most excitingly for thinking about problems in soft matter in general, or self-assembly problems, this opens up new ways of tuning kinetic pathways in complex systems by being able to selectively drive parts of the system away from equilibrium and do things more rapidly than would be allowed by equilibrium rules. So with that, I'd like to thank my group, in particular Fan, Youngik, and Jessica, who did most of the work that I showed you. Happy to take any more questions.
So just going back to the first part, within the framework of the toy model, if you have specific epsilons that then design a certain number of condensates, and now you fix the epsilons, and then you're given a sampling of the condensates, can you infer back something about the epsilons, at least in the sense of the low rank part?
Most likely, yes. So there has been a lot of work on using random matrix theory to try to connect only statistical properties of distributions to the types of condensed phases you can get in a multi-component system. So if you, and also in particular in systems that where you have low rank in matrices, it's usually possible to infer the rest of the matrix from only partial information, and there are a lot of theorems related to that. So I think putting all of that together, it should be possible to have some combination of sort of optimization approach and sparse experimental data to then infer what else must be happening within the framework of this model. Yeah, I think that's a really exciting idea.
Yeah, just one question on this random matrix approach, because I mean, I would expect that in random matrix that is level repulsion. So actually you have all the eigenvalues that are different from each other. You found here actually that most of them are equal. So what kind of random matrix are you using? With a lot of symmetries, with a lot of I mean, yeah, you see what I mean, right?
Right.
If you generate a random matrix, you're never gonna find two eigenvalues that are equal. So here you find most of them equal. So it's kind of what kind of model you use in the polymers.
In that part, right, where we had
when you find only four eigenvalues that are different out of.
Right. Yeah. So the specific use of random matrix theory there was just saying that in whatever calculation you do, whether a simulation or an experimental measurement, there's always going to be noise, right? And so if you want to distinguish what is noise from what is signal when it comes to these eigenvalue spectra, then if you know that you can independently measure the error on a couple measurements, then you can predict what the distribution of eigenvalues should be. So when I said they're all at zero, they're actually all very close to zero. And the ones I highlighted
Then
were
you
the ones that were
subtract, okay, you
clearly
subtract the
not
spectrum
bound.
from the random matrix, and the rest is the signal. Okay, okay,
Yes,
okay.
yes.
Okay.
And so in general, if you think about these biomolecular interactions, they're probably mostly low rank with some noise on top, and then you can use.
Yeah, I understood. Okay.
So sure. Go ahead.
I want to defend Mr. Gibbs. I think you were unfair to him because he didn't say anything about equilibrium or metastable or anything.
No,
He
of
said
course.
only two things. If several phases coexist, then pressure should be equal in all of them, and chemical potentials of all components should be equal. You want the same thing. Whether it's metastable or stable, no one cares.
Yes. So Gibbs is always correct. Right? That's the most important.
Thank you.
Right. That's
I
the most
want
important
to
thing.
hear
That's the most
that.
important thing. The key, yes. So the point of emphasis there is that these observations are in no way contradictory. However, is a question that almost always comes up. Why are they not contradictory?
Yes, so
The key,
it must mean that you ask different questions. then.
absolutely, absolutely. We're asking a different question from saying, how many phases do you have at a global equilibrium on a phase diagram where you have no linear dependencies of the interactions. The last part is a technical thing that you actually need to say. Gibbs phase rule only works if you have all of your chemical potential equations are linearly independent. He also knew this.
Another aspect of difference is that who are considered different species, different molecular species. In your case, different sequences are different. For Gibbs, those would be different species.
Absolutely. And
therefore, you have many species.
Right. So here, the distinction on when you have a new species is because its column in the interaction matrix differs from the other columns. Right? And again, this is where these like everything has to be specified with regards to some bounds or tolerances, right? So when we say let's make a low rank matrix, it doesn't need to have all other eigenvalues exactly equal to 0. They just have to be small enough that it doesn't matter for what we observe. Right? So when we say is it a new species or not, because biomolecules tend to have lots of chemical modifications and may be slightly different, the question is, are they distinct species in the sense that their pairwise interactions differ by more than some tolerance, at which point they behave sufficiently?
So I have a question for me, and then I have a question, I guess, for or from John Brady. So the one for me, I don't know where I'm going with this, but the delta mu you were using your non-equilibrium steady state process that leads to these fluxes on the interface. Does that entirely define what the entry production of that process is? Or is there something that you could quantify and maybe be informative about like how rapidly the condensate would form because we make the process more irreversible? I don't know if that's something you thought about. And then for or from John, that because you have this flux, then you might have some notional pressure around the particle. So then I was wondering whether all the work that has been done in the context of non-equilibrium condensation for something as simple as MIPS, whether all that framework would be useful to think about this in a way that is completely removed from the equilibrium thermodynamics language.
Yeah, great. Those are two fantastic questions. I should thank only you. Okay. So
you could have asked the same one better.
All right, so
maybe you already know the answer. That's why I didn't ask the question.
To the first one about the what you can learn by looking at entry production. Of course, it's physically very important quantity.
I'm not sure about that.
It tells you if you're at non-equilibrium, right? So
yes. So you know that from the flux. That's what I'm saying. One more does it give you.
Yes. It turns out in this case we have not been able to say anything new or more informative on the basis of that. So I
guess my point was that does the delta mu determines the entirety, or there is some cooperative effect on the surface that controls what the net entropy production is?
So we're able to predict everything on the basis of the parameters that I showed you, right? So these are the control parameters, the delta mu being the key one. And if you know that, you're able to predict what happens here in terms of the theoretical predictions for what the nucleation rates are going to be, what the surface tensions
But does it tell
are
you
going
what
to
the
be.
total entropy production of the assembly process is?
I mean, in the sense that you can go and calculate this in the simulation,
Okay, so you need simulation.
Getting at that analytically is possible for the simplified theory, but for the full thing, it's something we have to do in simulation. One of the main reasons that it doesn't tend to be that useful is that it's possible to drive the system in ways that do not affect the nucleation rate. And you can have cases where you have vastly different entropy production in the two different phases, and yet see no obvious or no measurable changes to interfacial properties or nucleation rates. So simply seeing differential entropy production at different points in your system doesn't necessarily mean that you could easily distinguish macroscopic observables such as capillary fluctuations from what you'd have in equilibrium. So to the second question about pressure. So yes, this is a crucial consideration whenever you deal with phase coexistence. You first have to say what you mean by having phases in coexistence, right? So these are systems that are not in equilibrium, so therefore you cannot invoke equal temperature, equal potentials and pressures, right? But you can still invoke the what they mean in practice, right? So you should have no net particle flux between the two phases. If they're in coexistence, you should have no net motion of a planar interface. If I have liquid here, vapor here, and flat interface, and if I'm in coexistence, it shouldn't be moving here like this, right? And so it's not equal pressures necessarily, but you can find where the conditions under which the interface doesn't, and define that to be the equivalent of mechanical balance condition.
Right.
So we haven't yet figured out how to directly predict that in terms of mechanical balance results, but it's exactly what we have to do to define a supersaturation. Saying how much does the interface move into the other phase because there's some sort of non-equilibrium thermodynamic-like driving force to go that way.
That's a different question. So in the end about the effect of the nucleation rate and the out of equilibrium that you only had 2 kT and you're quite different. Can you turn it around and say that if I observe the rate of formation, and then infer how far the equilibrium might must be in order to get that, and compare that to other measures one might have of how out of equilibrium the system would be?
Yes. So absolutely, that's one of the main things we wanted to do when we started this, is to say, with simple models, when can you actually see things in a way that would be easy to detect experimentally, such as interfacial tensions, and then infer back. So I think the simple answer is, if you see a significant change in the interfacial tension in these systems, while you don't change the temperature, then you must be out of equilibrium, at a non-equilibrium steady state. That's something that we can show for sure. But if you don't see that, it does not mean that you're necessarily at equilibrium. It's possible to be out of equilibrium and see no reduction.
Could you infer, even from the measurements of observations of the actual phase separation taking place in the neural system, that if the nucleation is responsible, that's happening much more quickly than one would have predicted, and therefore get a measure of how far
Indeed.
way in.
Yes, so it should be possible. I think the difficulty is that you need to know the equilibrium, equivalent equilibrium behavior to be able to compare that, because I don't know if it's fortunate or unfortunate, but a lot of the scaling laws that you'd expect from classical nucleation theory, and lots of other aspects of these non-equilibrium phase transitions. A lot of the scaling laws look the same. So you actually need to measure material properties to be able to say the difference.
I was going to follow up on that, because like some of those steady states could be clock cycles, and so like the timing reactions that chemists like, and so that made me wonder if biology actually makes clocks like that by having different compositions of droplets that then interact. Some go out of existence, some come into existence in a clock-like way. Is that some sort of mechanism that biology already uses?
Probably.
So
there are certain
in the
cases,
body of the cluster, you don't have fuel.
right? Yeah.
Right.
So there are cases that are not that I'm aware of, and again, biology that I'm certainly more things I'm not aware of, right? But there are definitely cases where things related to say cell division involve the formation of these non-equilibrium droplets that then dissolve, and I wouldn't say it's a clock necessarily, but the timing of these events is synchronized by these things forming and dissolving. And so there are a couple examples of that.
I forgot to ask something about surface tension. You extracted this quote-unquote surface tension from mean square average fluctuation.
Yes.
Did you check the Q-dependent spectrum of this fluctuation? Is it consistent with having local surface tension?
Yes, that's another example of a scaling that doesn't change. But it's more that all of everything's just shifted.
So the only thing that changes is the overall strength of the surface tension.
Yeah, the overall, so basically taking a variance and you're decomposing it into modes, and distribution across those modes is the same as what you'd expect at equilibrium. The overall variance is higher.
No additional questions. Let's thank the speaker again.